inside every single one of your cells right now. This tiny, elegant trap is being set over and over. It's a process that makes sure your body can hold onto its most precious fuel. So today, we're going to pull back the curtain on the secrets of the sugar trap.
I mean, really think about it. Every cell in your body needs energy to do its job, and its absolute favorite food is a simple sugar called glucose. But here's the thing, that fuel is slippery. So how does your body stop it from just, wandering off before it can be used?
And this is the heart of the problem, the cell membrane. It's not some locked fortress. For a molecule like glucose, it's more like a revolving door. It can wander in, sure, but it can just as easily turn right back around and leave. And for something that needs a constant supply of energy, well, that's not exactly a great setup, is it?
To solve this, nature came up with something incredibly clever. It doesn't build a better wall or a stronger door. Instead, it changes the glucose molecule itself, so it just can't fit through the exit anymore. It sets this brilliant one-way chemical trap.
The fancy name for this molecular mousetrap is phosphorylation. Now, it sounds super technical, but the idea is actually pretty simple. You take a phosphate group, which is basically a little cluster of phosphorus and oxygen atoms, and you just stick it right onto the glucose molecule. That's it. But that one tiny addition changes absolutely everything. And the results? It's simple, but man, is it powerful. The second that phosphate group clicks into place, the glucose is locked in. That revolving door just slams shut. But how? How does this little chemical lock actually work?
Okay, this is the really genius part. That phosphate group isn't neutral. It carries a strong negative charge. And the submembrane is basically designed to repel charged things. So our glucose, which could once waltz in and out, is now trying to get past the velvet rope with this big, flashing, negatively charged sign on its back. The bouncers at the membrane door take one look and are like, nope, sorry, you can't leave. And just like that, it's trapped inside the club.
All right, so we get what the trap is. Now let's dig into the how. What does the actual chemistry of setting this trap look like? If you were to do this in a lab, it's all pretty straightforward. It's a classic bit of chemistry called esterification. As you can see from the source notes, you basically mix glucose with phosphoric acid, they react, and boom, you get our target molecule, glucose 6-phosphate, and a little water. Easy peasy, right?
But hold on a second. If you look closely at that glucose molecule, you'll see it's covered in spots where this could happen. 5 different alcohol groups. So why does the phosphate specifically and always target the carbon atom way out at position number 6? Why not number 1 or 3 or any of the others?
Well, there are 2 big reasons, and this diagram shows them perfectly. First, accessibility. I mean, just look at it. That group on carbon 6 is just hanging out there in space, this easy target that's not all crowded by the other atoms in the ring. And second, there's reactivity. Chemically speaking, that particular type of alcohol is just more eager to react. It's the path of least resistance.
But that's chemistry in a beaker. Our bodies, way more sophisticated. The real deal biological version is a total masterclass in control, using specialized tools to get the job done perfectly every time.
And here's the crucial difference. Your body doesn't just have bottles of phosphoric acid floating around. Instead, it uses a very special molecule to hand-deliver that phosphate group. And it doesn't leave the reaction to chance. It uses a powerful molecular machine to make sure it happens instantly and flawlessly. That special molecule is ATP. You've definitely heard of it. It's the universal energy currency for all life on Earth. Think of it like a tiny, charged-up battery. But for this specific job, it's not really providing energy. It's acting as the phosphate donor. It's a delivery truck that willingly drops off one of its 3 phosphate groups right onto the glucose.
And the machine that makes it all happen is an enzyme called hexokinase. Enzymes are like these tiny biological robots, and hexokinase is a real specialist. Its one job is to grab a glucose molecule and an ATP molecule and perform that phosphate handoff with lightning speed, the very instant that glucose crosses into the cell. Talk about efficiency. The whole thing is like this perfectly choreographed 4-step dance. First, hexokinase binds to both glucose and ATP. Then, the enzyme itself literally changes shape, squeezing them together into the perfect orientation for a reaction. This sets the stage for the attack, where an oxygen atom on glucose basically snatches that final phosphate right off of ATP. And finally, the old bond on ATP breaks and a new one forms on glucose. The trap is officially set.
So, the glucose is trapped. Mission accomplished, right? Well, that's actually only half the story. There's another equally important payoff to this whole process. I mean, you might be thinking, is all this complicated machinery, all this ATP being used, really just to lock a single sugar molecule inside a cell? It seems like an awful lot of work for just that. And the answer is a firm no. There is a second reason we do this, and it's all about getting the glucose ready for what comes next, unleashing its power. The perfect way to think about this second job is like a spark. By adding that negatively charged high-energy phosphate group, we're not just trapping the molecule, we're also making it unstable and reactive. It's like turning a stable log into kindling that's been soaked in lighter fluid. It's ready to ignite. In biochemical terms, this is called priming. Phosphorylation is the investment step. The cell actually spends one molecule of ATP to prepare the glucose for the main event, glycolysis. This is the big metabolic pathway that's going to shatter this primed sugar molecule and begin to harvest all of its energy. So let's bring it all home. This one simple reaction, the very first thing that happens to sugar when it enters your cells, does two vital jobs. First, it's the trap, making sure your fuel supply can't just walk away. And second, it's the spark that primes that fuel, getting it ready to burn. It is a masterpiece of biological efficiency. Which, of course, leaves us with one final question. The fuel is trapped, the fuse has been lit. So what happens when that energy finally gets unleashed? Well, that's a story for another time.